We're going to look at what kind of organisms cause disease. How do we know what organism did this? So basically this part looks at how do we go about establishing pathogenicity of something? How do we define it, and how do we define it as far as the scientific method goes? We have to be able to test for pathogenicity. The tests that we use were developed by a gentleman called Robert Koch back in the late 1800s. He came up with a concept of being able to apply scientific method to determining what causes a disease. We call these Cox Postulates. The first one, the suspected causative agent must be found in every case of the disease and absent from healthy hosts. We're looking at an association between a pathogen and a host where we can find the pathogen when we have disease, but we don't find the pathogen when we don't have disease. Therefore, the association goes back to the observation. Next, we must be able to isolate the agent in pure culture and grow it outside of the host. This goes back to controlling the variables. We find one agent, we grow it outside of the host, we therefore have it in a pure culture, and when we have it in a pure culture, we can determine what it is and verify that we are only using one particular organism. Third postulate says, when the agent is introduced into a healthy susceptible host, the host must get the disease. We then take that material, which is pure, and we apply it back on the host. The host will then develop symptoms out of this. Therefore, we are looking at being able to close the loop and say, hey, this particular material caused that particular disease. Robert Koch came up with these three parts. Later on, the check that basically says the original agent must be re-isolated from the disease experimental host. When you do this, you are then able to compare what you remove from the host this time with what you put into the host the first time. If the two of them coincide, you have now verified scientifically Cox postulates. This is to be able to prove that something caused a disease. When we look at these, there are some exceptions to Cox postulates. These become very important. Some pathogens cannot be cultured in vitro. We can't really grow viruses in the total absence of other organisms because viruses only grow inside of cells. There are certain fungi that need a host to be able to complete their life cycle. We can't really isolate these in pure culture. We have to deal with them a little bit differently. Some diseases are caused by a combination of factors. I want to talk about this in humans is HIV. HIV is a pathogen that does something that then allows something else to happen, that allows something else to happen, and this is what makes you sick. So when we deal with this, we've got a combination of factors. It's not a simple A or B. These are important in Cox postulates. Then you've got the third one that cannot be satisfied within ethical boundaries. This is the part about inoculating a healthy, non-diseased host. This past year we had quite a problem with Ebola worldwide, and you really can't go out and take the Ebola virus and inoculate it into somebody to see whether or not it's going to work, because chances are you're going to end up killing them. Therefore, this violates any sort of ethical boundary we use. Question comes down to it. Does this only deal with human pathogens, or could we look at other pathogens as well? For the most part, in plant pathology, we have to complete Koch's postulates in order to be able to verify that something is actually a pathogen for a particular host. When we look at pathogenicity, Koch's postulates become one of the ground rules, becomes one of the very important parts that we have to be able to deal with as far as proving pathogenicity.